Good day, everyone. We are from Group 1, and we'll be reviewing an article titled Combining Genetic Algorithm with Machine Learning Strategies for Designing Potent Antimicrobial Peptides. This is the overview of our video presentation today. So, let's start with the introduction of our topic today. First of all, the main issue that is discussed in this article is about antibiotic-resistant microbes. As we all know, recently the antibiotic-resistant infections are a growing public health concern. As a solution, the scientist came up with one answer, which is the antimicrobial peptides, AMPs. It is effective against antibiotic-resistant microbes and has various mechanisms of action. So how are the scientists planning to create this AMP? They plan to use machine learning to design and create the AMP. But there are some problems regarding machine learning. As we all know, in recent times, machine learning is widely used in solving complex problems. However, a part of machine learning called the deep learning method can make illogical connections. This is because the lack of transparency in deep learning methods can limit the knowledge gained from machine learning. To overcome this problem, the scientist came up with a solution called Explainable Artificial Intelligence, XA. It aims to address the lack of transparency and problem in deep learning. In this method, they use transparent decision approaches like the rule induction method. Specifically in this article, they use the rough set theory, which can remove the illogical connection. Rough set theory helps understand the relationship between input and output labels. This article mainly discusses about how to create the AMP using machine learning. Designing the AMPs requires considering physical-chemical properties and machine learning approaches. To create the AMP, plenty of methods and techniques are employed. First of all, they use computer-aided molecular design, CAND, to design the molecules. Secondly, they use the rough set theory. It can be used to classify peptide sequences based on their antibacterial activity. Next, the combination. The rough set theory is combined with a genetic algorithm search in CAMED to design the antimicrobial peptides. The ultimate aim is to design antimicrobial peptides against specific targets. The method used in this study involves two main approaches. The first one is active peptide customization using rough set theory. First of all, the rough set theory identifies relevant property value intervals, which differ between classification labels. They use this rough set theory to determine the intervals of summary sequence property values that are calculated from the amino acids' chemical properties using the Aindexon database. This can be used to separate active peptide sequences from inactive ones. There are two rules used in the selection process, which are MLEM2 and CLAMLEM2. In order to design the AMP, the main thing that is observed is the ability of the amino acids to form alpha, helix, and coil structure. The method works in iterations, and in each iteration, they use the rules to predict if the new peptides are active or inactive. The next method is to discover the customized peptide sequences using codon-based genetic algorithm. This method is used to find peptide sequences with specific design targets. First of all, the previously known antimicrobial peptides are ranked based on their targets. Then, the top 25 of Canti5 candidates are selected for mutation and crossover. These candidates are represented by their DNA codon. The represented DNA codon is selected and combined to create new peptide sequences. On top of that, motifs, which are the repeating patterns in the DNA codon, are captured by MLEM2 rules and are used to generate new sequences. The highest scoring unique and predicted antimicrobial peptide sequences are copied between generations. Finally, the highest scoring sequence across generations is considered as the final solution. The next step in the process of creating the AMP is the peptide synthesis itself. The peptides are synthesized using an APHEC-Focus Exipeptide synthesizer. Lastly, to measure the efficacy of the newly created AMP, the scientists decide to use the zone of inhibition tests. In this test, Staphylococcus epidermidis is used as a test strain. The antimicrobial peptide candidates are tested using a zone of inhibition assay. Firstly, the Staphylococcus epidermidis is cultured on the agar plates. Then, the solubilized peptides are pipetted onto the plates. The plates are incubated, and the zone of inhibition is measured to evaluate the antimicrobial activity.
Here comes to the result part. First result is regarding the generation of increased sequence diversity through codon basis. As mentioned in the methodology part before, the researchers explore the use of codon representation to generate novel antimicrobial peptide sequences. They introduce mutations through substitution, insertion, or deletion of a single DNA base and employ crossover techniques to combine subsequences of predicted AMPs. By using the codon representation, they aim to take advantage of reading frames and increase the diversity of generated sequences. The figure at the right side shows that the codon representation leads to higher variance in fitness scores compared to non-codon representation, but it ultimately reaches similar maximum and mean fitness levels. Next, in order to generate a new peptide sequence, Emlem's rules are used in this screening process. Only the sequences which adhere to the MLEM2 rules for antibacterial activity pass this screening. Also, the codon representation method that yields fewer sequences with increased fitness score variance will pass the screening. The researchers evaluate the diversity of screened sequences by comparing the codon and non-codon methods in terms of parameter coverage. When the coverage of the parameter space is higher, it means that the selected sequences represent a broader range of values or combinations of parameters. This indicates that the designed peptides have diverse properties and cover a wider spectrum of potential antimicrobial activities. We can see from the figure at the right side of this slide, it reveals that the codon representation method offers better parameter coverage, primarily because it incorporates sequences resulting from frame shifts. In our previous work, we focused on the forward problem of designing antimicrobial peptides using computer-aided molecular design, COM. Now, we address the reverse problem by introducing our codon-based genetic algorithm, PBAY, method. We start by sorting known antimicrobial peptides based on their scores and design targets, focusing on sequences that are easily synthesized using the FOMC protection method. To simplify the synthesis process, we avoid sequences with cysteine residues and prioritize sequences with lower aggregation potential. Additionally, we target cationic peptide sequences, which are known to exhibit antibacterial activity. To increase the probability of peptide activity, we incorporate multiple clone MLEM2 rule categories as design targets. The result of the antibacterial screening experiment using Staphylococcus epidermidis is that they found out 1 out of the 3 antimicrobial peptides chosen through their genetic algorithm demonstrated antibacterial activity. As a reference or positive control, they included a peptide called APE1404, has highest scoring sequence and which known to have activity against gram-positive bacteria. They compared the aggregation potential of AMP2, one of the selected peptides, with that of H1404. The average aggregation potential of AMP2 that has the value of -0.2 was lower than that of H1404, which has the value of 0.26, indicating that AMP2 had a reduced tendency to aggregate. Despite this difference, AMP2 still showed antibacterial activity against Staphylococcus epidermidis. Based on the result given, we can know that by using the codon representation, the variation can be increased. This is because, in order to design peptides, a genetic algorithm is employed, utilizing a DNA codon representation that leverages reading frame shifts. By introducing frame shifts through single codon mutations, we enhance the diversity of generated peptide sequences. Although using the codon representation reduces the number of predicted antibacterial sequences, it leads to greater diversity and wider coverage of target score dimensions. We filter out non-antibacterial sequences, so the increased generation diversity focuses on predicted antimicrobial peptides. This is the first time a genetic algorithm for peptide design has utilized reading frame shifts to generate novel sequences. Next discussion part is about the combining antibacterial classes. Each rule in CLON LEM2 represents a specific set of characteristics that can differentiate antibacterial peptides from non-antibacterial ones. The number of CLAN MLEM2 rules satisfied by a peptide serves as an indicator of its potency or efficacy in exhibiting a broad spectrum of antibacterial activity. This is because different rules might represent different mechanisms or ways in which the peptide can act against bacteria. By combining these different sequence features can help create a more effective antibacterial agent. In conclusion, application of transparent machine learning algorithms, particularly rough set theory, in conjunction with an evolutionary search method, proves valuable in the design of antimicrobial peptide sequences. To enhance the targeting of antimicrobial activity against antibiotic resistance, microbiome dysbiosis, and immune system suppression simultaneously, our proposed computer-aided molecular design approach enables the customization of antimicrobial peptides with specific desired properties and strain. Specificity. This approach has yielded successful outcomes in discovering new antibacterial peptides that are more convenient to synthesize compared to existing ones. Furthermore, it has improved antibacterial effectiveness by integrating multiple activity rules derived from rough set theory. The methodology involves utilizing rough set theory as a model for quantitative structure, activity relationship, facilitating peptide design, and employing a codon-based genetic algorithm to identify antibacterial sequences. Experimental results assessing the activity against S. epidermidis in a laboratory setting provide supporting evidence for the efficacy of the transparent machine learning approach. The developed technique not only offers increased sequence diversity, but can also be expanded to encompass diverse pathogens. These are the references that we used to do the article review. That's all from our group. Thanks for watching and have a nice day.